So I'm going to tell a story today about motility of algae through day-night cycles. And this is not to say that other microorganisms are less important, but today I'll just talk about algae. And the work was carried out by Diel, who was my PhD student in Cambridge and is now at the Weizmann Institute in Israel, and it was a collaboration with contributions by Yuri Kota at the Cavendish Laboratory, Emma Sylvester, who is now at the Structural Biology Unit in Oxford, and Kyriakos Leptos at DAMT in Cambridge. And it was funded by the Winter Programme for the Physics of Sustainability. So a bit of motivation for algae. So algae widespread in the environment. You can see here a picture of these coccolithophore algae. So the algae that have a calcium carbonate shell, and they form these blooms here seen between France and the UK. And so algae in the environment are responsible for fixing about 50% of atmospheric carbon, and for comparison, land plants fix about 25%. So they're really important carbon fixers and play a critical role in climate regulation. Over sort of geological time, these coccolithophores grow and then die, and the shells can sink, and over geological time form sediment, which is results in these chalky white cliffs of Dover. Another useful carbon sinking that algae do is in biotechnology. You see here these raceway ponds in Israel, which you'd expect them to be green, but no, algae are not always green. When they are stressed, they produce pigments, like this alga beta-carotene, which produces this alga, Diurnally salina, which is a marine species which produces beta-carotene, which is a valuable pigment. So you can grow algae in these ponds to produce useful products, and that's another motivation for studying them. So this same Diurnally salina, of course, we are interested in algae that swim. So this Diurnally salina has two flagella, which it beats to swim at low Reynolds numbers. It has interesting responses to flow. So here, a suspension of Diurnally salina has been exposed to oscillatory flow, and we discovered that these trajectories align when you hit particular values of amplitude and frequencies, so the trajectory exhibit resonance. Algae also respond to light. So here on the bottom left, you have a suspension of Chlamydomonas reinhardtii. And when the arrows go on, there's a strong light from the right. And you'll see that the algae don't like that, and they move away from the light. And this is called negative phototaxis. So algae have these interesting response at the microscale, and at the macroscale that results in weird fluid mechanics. So a prototypical example is this gyrotactic focusing, which is discovered by Kessler. So if you have a swimming alga which is bottom heavy or has anyway a center of mass offset from the center of buoyancy, then the torques that act on it due to gravity and flow shear, if you put it in a shear, reorient the cell so that in a downwelling pipe flow, as shown in this picture, the cells are biased to swim towards the center of the flow and form a plume in the middle. Now this is no longer weird, but when Kessler in the mid-80s observed this, he was quite surprised to see this sort of spontaneous pattern formation in a fluid of swimming algae, and eventually he went on to explain it. Kessler was working on this because he was interested in desalination, and he started working with Diurnally salina because it produces copious amounts of glycerol to protect itself from osmotic shock. And so he was messing around with Diurnally salina, and he saw that it formed patterns. And then he actually happened to be in Cambridge, where there was a culture collection of algae, and he found other species such as this Chlamydomonas augusta, which form patterns. So I've been involved in exploring, in collaboration with Martin Bies and others, the consequences of this gyrotactic focusing. So we looked at how cells disperse in a pipe. So normally, if you have a passive solute, it will just be advected by the flow, and it would diffuse in the cross section of the pipe. And the solute will be like a Gaussian that spreads. But what we explored is, if you have these gyrotactic focusing plume structures, how do cells disperse in that kind of situation? And we found that a suspension of algae has a non-zero drift above the main flow. So a passive tracer is just carried passively by the flow, but a suspension of swimmers has a non-zero drift. And I did some experiments where I imaged suspension of Diurnally salina drifting in a pipe flow. And I measured the drift against the Pecklin number of the flow, which is a measure of how strong the flow is. And I compared these non-zero drifts for different values of the cell concentration, finding reasonable agreement. Other recent examples of this sort of weird fluid behavior. Durham et al. Found that if you have a vertical flow, you can have these plume structures. If you have a horizontal flow, you can have this gyrotactic trapping. So if where the shear is stronger, the cells tumble and they get trapped in a narrow band of shear. And this was posited to be relevant to the formation of thin layer structures in the ocean. So this is also a really nice example of how a suspension of algae which responds to light can behave. So here you have a light shining in a suspension, and you'll see what happens. As the light shines, it concentrates the cells and generates an instability, causing the cells to sink. So this was work by Arrieta et al. And Marco Polin's group. And then another example is this active rheology. So if you do a rheology with swimming algae, the effective viscosity is different from that of a passive suspension, and this is because the algae are pullers, so they exert a stress on the fluid, which reduces the viscosity. So to describe these suspensions in the dilute limit, so maybe excluding the last example of active rheology, you can use a continuum model, and these were developed in the 90s by Pedley and Kessler. And the model has the form of a conservation equation for the density of cells. So you have an advection due to the fluid flow, an advection due to swimming, and then cell diffusion with an anisotropic diffusion tensor. And then you just have the Navier-Stokes equation for the flow. So just the flow variation changes with a pressure gradient, a stress, which can include axis stresses due to swimming, and sinking. So the cells are slightly denser than the surrounding fluid, and so they generate a source in the fluid flow. So, for example, if we consider the conservation of cells, this has two active transport measures: the swimming velocity and the diffusivity tensor, and these depend on swimming parameters. Clearly, the swimming velocity depends on the mean swimming speed, and the diffusivity depends on a ratio of the mean swimming speed squared divided by the rotational diffusivity. So these models are reasonably successful at describing bioconvection patterns, plumes, photobioconvection structures, but they need to be parameterized from experiment. And I've made the example of continuum models, but the same applies for individual-based models. So, for example, we can use microscopy to obtain these parameters. This is a technique called differential dynamic microscopy, which allows you to use the fluctuations in the image due to the motility of organisms to infer swimming parameters such as the mean swimming speed, the width of the distribution, the amplitude and frequency of the beating of, in this case, Chlamydomonas reinhardtii. But if you look at the literature, so what is the speed of Chlamydomonas reinhardtii? Well, everybody knows it's around, everybody in the field will say, well, it's about around 100 microns per second. An order of magnitude, you'd be doing okay. But if you search the literature for measurements, you find quite a range of values. Now, these are different strains, so you could imagine, well, maybe it's due to the fact that it's a different strain. Notice that in this case, the strain wasn't even mentioned, for example. But sort of, for example, if we pick the same strain, we have different values of the swimming speed. And if we look at the culturing conditions, they're more or less the same, but not quite. So for example, this TAP it stands for tris acetate phosphate, and it's if you add it to the media of Chlamydomonas, it can grow on the acetate by fermentation, as opposed to growing photosynthetically on the light. So it's a way of speeding up the growth of Chlamydomonas. It's convenient, it grows quicker. But the question we wondered about is how do these growing conditions affect the swimming speed of Chlamydomonas, and in particular, how do day-night cycles change the motility parameters? So the Chlamydomonas is a photosynthetic alga. So it's like a single-celled plant, and it grows during the day with the light, and then it divides on the onset of the night, and then it grows again and it divides. So if you put it in a light-dark cycle, the day-night cycle in the environment, then this circadian variation gets synchronized to the light and dark. And so we refer to the time as the Zeitgeber time. So that's German for time giver. So the light is the time giver. The light is what sets the clock of Chlamydomonas in terms of its growth and development. So you see the pictures here. The algae grow during the day, then they divide at night, and so on it goes. So we set about studying the variation of the statistics for Chlamydomonas. So here I want to remind you that this was the work of Diel, who undertook a very challenging set of experiments where she measured a lot of parameters of Chlamydomonas growing and swimming. So in particular, she counted and sized the cells using a Coulter counter. She inferred the motility stats from tracking using a vertical microscope in a dark room with a red light. So that's to avoid phototaxis, and the vertical microscope is to parameterize the gravitaxers, as we'll see in a minute. She counted the cells that didn't swim using a horizontal microscope with the light as above. She inferred the density of the cells from the sedimentation of immobilized cells, and she also assessed the cell shape and condition. So all these steps were done for different points in the day-night cycle, and they took maximum 1.5 hours. So for example, this is the density of cells as a function of this Zeitgeber time. And you can see that during the day, the density is constant. And then as the cell starts to divide, density goes up. So I should say that the synchronization process involves growing some cells and diluting them, and then growing them again and diluting them until in day-night cycles, until the cells are synchronized to the day-night cycle. The dilution makes sure that you end up with cells that are all the same size. And so you can see in these pictures at the bottom, at the Zeitgeber time 1, and then at 13, the cells have got bigger. And then at 24, you can see there's more cells. So the size, the volume, so this is a plot of the volume of the Chlamydomonas cells as a function of time. And the volume grows, and then it reduces as the cells divide, and so forth. So the motility statistics were acquired by using tracking microscopy. We didn't use differential dynamic microscopy or holographic microscopy, or one of these techniques which are 3D, for historical reasons. And so we estimated the mean swimming speed by approximating, by assuming that the distribution of cells was strongly peaked, so that we could approximate the square of the mean with the mean of the square, and also the speeds in the horizontal plane were the same. So the distribution was quoted by Fujita et al. To be strongly peaked. So then we measured the swimmer fraction. So this is the number of swimming cells divided by the total, as inferred from imaging. Then we measured the bias parameter. So this is a measure of the ratio between the rotational diffusion time and the gravitactic reorientation time. And it was shown by Pedley and Kessler that the average of the upward swimming speed divided by the mean swimming speed is related to this function of this parameter lambda. So we could, by measuring the speed in the z direction, we could, and the mean speed, we could infer this parameter. We then measured also measured the rotational diffusivity and the gravitactic time. So this was done by measuring the autocorrelation of the swimming direction of Chlamydomonas, and because of the measurements were projections, we used simulations to infer the 3D value. We also measured the mass density from sedimentation experiments, as said before. So this is a plot of the swimmer fraction as a function of the time in the day-night cycle. And I want you to consider that this plot is periodic. So as you go out in the day here, you come back at the beginning here. And so you can see that the swimming fraction is constant during the day, and then it falls just before nighttime, and then it goes up again. So this was not a surprise, because it's known that when Chlamydomonas divides, it pulls in its flagella. So this is a nice little video from the Chlamy lab in Canada, and you can see that the flagella are resorbed prior to division. And this is because the machinery of the flagella is actually required for division. So the cell is doing a nice little piece of recycling. And so as the cells stop swimming, the fraction of swimmers goes down. It doesn't go down to 0 because there are always some cells that are swimming, that these processes don't happen all at exactly the same time. But then was the surprise. So Dee measured the mean swimming speed, and she found that it is constant at a value of about 80 microns per second during the day, but at night it pops up to around 140 and a maximum of 160 microns per second. So Chlamydomonas appears to swim faster during the night phase, which, as far as we know, had not been observed before. What had been observed before was that the Phototaxis of Chlamydomonas showed day-night variations. So these curves represents the strength of the response of Chlamydomonas in experiments by Bruce. He's shown some light on a suspension of Chlamydomonas, and the accumulation changed the transmitted light, and these lines are a measure of the strength of a transmitted light on a detector. And also Chlamydomonas was shown to be chemotactic to ammonium, and this chemotaxis also seemed to be here, for example, stronger during the night, which maybe is due to the swimming, but these experiments, I think, will be really nice to do them again. So so it'd be nice to measure this beat frequency, and this could be done directly by other techniques, but with the technique that we use, we were not able to measure the mean frequency directly. but by using an argument about the power balance, it is possible to infer the frequency as well. So as Chlamydomonas swim, the power is just the work per beat times the mean frequency, and this is equal to just the drag force times the mean swimming speed. The drag force is just given by Stokes' law, where Cf is a form factor, and if you insert this expression for the force into this first equation, you see that the power scales like the swimming speed squared, which is what would be expected and was measured for Chlamydomonas by Guasto et al. In 2010. Then a Japanese group, the Fujita et al., they measured the mean swimming speed as a function of beat frequency by using different mutants of Chlamydomonas, which beat slightly different frequencies. And so it's possible to obtain a relation between the swimming speed and the beat frequency, where the swimming speed doesn't go to zero as the frequency goes to zero because of limitations due to the tracking that these measurements were carried out with. So what we did is we fit the data by Fujita et al. And this allows us to infer a frequency from the swimming speed, and this is what is plotted here. So the frequency changes with the time of day, and it seems to sort of broadly decrease during the night and then flatten out during the day. The magnitude of the frequency, if you compare for other measurements with Chlamydomonas, so this sort of late daytime, early nighttime value is 60 Hertz, which coincides with measurements by Wan et al. For the same strain, but just with the caveat that this was grown on tap media rather than the acetate-free media that we use to ensure the synchronization. So for sperm, the beat frequency was recently shown to grow with the concentration of ATP. So they had a demembranated urchin sperm, and they could calibrate. They could measure the ATP concentration from the fluorescence of an ADH biochemical reporter, and by doing that, they were able to plot the beat frequency as a function of concentration of ATP. As you would expect, the beat frequency of sperm increases with increasing ATP concentration. And at the time where we wrote the paper, this is all that was available in the literature. They did this group also studied Chlamy, but they didn't demembranate or study the ATP concentration dependence for Chlamy. But recently, a paper has come out where they've also shown that the beat frequency of Chlamydomonas increases with increasing ATP concentration. Now, the caveat is the way they did this here is they demembranated Chlamydomonas, leaving the axonemes, which drive the beating, intact, but the cell is dead. And then they added the ATP, and it caused the dead cell to beat, and they established that the beating was similar to live cells. So it comes with that caveat. Anyway, it seems reasonable that the beat frequency of Chlamydomonas should increase with ATP. And so where does that ATP come from? So there are various pools for ATP in Chlamydomonas. There's ATP in the cytosol, there's ATP in the chloroplast, there's ATP produced by the mitochondria, and ATP also is produced in situ in the flagella. You can see from so this diagram plots mRNA abundance against time as a function of day-night cycles, and you can see that the transcription of the ATP synthase in the chloroplast drops as the night comes in, and the mitochondrial ATP rises and then drops during the night. So this is just to say that when the cell is not doing photosynthesis, then the chloroplast ATP is not going to be available. But during the night, the cell does respiration, and the mitochondrial ATP. So if you take our frequency, diagram and you assume that the high frequency corresponds to high ATP and low frequency corresponds to low ATP, and then you can hypothesize that the ATP is maybe dropping during the night, and that might make sense. So Chlamydomonas grows during the day and stores carbon and energy in carbon compounds, which are then burned during the night by respiration. So it could be that what we're seeing here is a decrease of the ATP as the nighttime progresses. I should say this, the ATP here is the ATP that is available to the flagella. And in terms of the ATP budget, it is used for growth, division, maintenance, storage, and swimming. So during the day, you have growth, and you have maintenance, and you have storage. And during the night, you have division. You always have maintenance, and all the time you have swimming. But the question is, how much ATP is available for swimming, and do our results imply that there's more ATP available at the end of the day because it is not used for other processes? So I think these are open questions. So then it's interesting to explore the consequences of a variation in the motility parameters as a function of time of day. So how much do your results in the lab depend on the time of day if you've got cells growing on a day-night cycle? So here, as an example, there's predictions from a model of gravitactic migration in a capillary. So you imagine you have a capillary full of Chlamydomonas, and these algae are gravitactic, so they're reoriented by gravity to swim upwards. And so the conservation of cells has contributions from swimming and cell diffusion. And you can use a stochastic microscopic model to infer the mean swimming direction. So this average of P is the mean swimming direction, which is upwards, and anisotropic diffusion. So it's a diagonal tensor with these components, horizontal and vertical components. And these factors all depend on the bias parameter, which is the ratio between the timescale for rotational diffusion, how the cell is reoriented as it swims along, and this gravitactic reorientation time, so how long it takes a cell to reorient to the vertical. So we can solve this conservation equation for the steady state and no flux boundary conditions at the top and at the bottom of the capillary. So if you do that, you just end up with this differential equation. And if you solve that, you have a vertical distribution, which is just an exponential. And the exponential has a characteristic length scale, which depends on the ratio of the swimming speed to the rotational diffusivity and a function of this bias parameter lambda. So this is like a sort of inverse atmosphere. You know, the atmosphere has an exponential distribution, and this Chlamydomonas is upside down. It's more concentrated at the top. So Michael Barry in the group of Roman Stoker for his PhD measured this distribution for the Alga Heterosigma Hakashiro, and he found indeed that there was an exponential distribution. Now these algae were grown with continuous illumination, so light was on all the time. So the question is, if you have a day-night cycle and you're sampling from that day-night cycle to measure this distribution, what are you going to get? So we can take the model that I just presented and use parameters from the parameters that D has measured. So for the speed, we take some values during the day and during the night. You can see that the rotational diffusivity does not have such a clear trend as the swimming speed in terms of our result. And the bias parameter doesn't seem to vary too much as well. Anyway, we can take these parameters and make predictions. And so here is plotted the vertical coordinate in the capillary, and as a function of the concentration of cells divided by the initial background value. And you can see that the swimmers distribute more broadly during the day than at night in the vertical direction. So another, so this was a sort of lab example, but an example where you get a day-night cycle with swimming migration of cells is the so-called Diel migration in lakes. So this is the species Chlamydomonas, but it's not Chlamydomonas reinhardtii, which actually lives in the soil, in Lake Limiton in Finland. And plotted is the distribution of these algae with depth as a function of time of day. So you can see that at 5 p.m., which I think is more or less, yeah, 5 p.m. where it's still daytime, the algae are sort of distributed on the surface, and as the night progresses, they become more or less uniformly distributed along the water column, and then as the day approaches, again they distribute themselves close to the surface of the lake. So it is thought that algae do this to take advantage of nutrients at depth and light at the surface. So compared to an alga that can't swim, which is more or less stuck at the surface, or has to do things like changing its buoyancy to migrate up and down the water column, motile species are able, through biased swimming, for example, gravitaxers, are able to take themselves up and down the water column and optimize their nutrient uptake, and also possibly avoid predation. I think is known that the zooplankton come to the surface around in the middle of the night. So these are not completely serious calculations. I'm just taking the values that we measured, and I'm evaluating the time that it would take to traverse a meter. And so there are speed values. So I have a night and a daytime value, and you can see that you can do a meter in 2 hours with our nighttime value, and you can do a meter in 3.5 hours with our daytime value. So I don't want to take these estimates too seriously, but this is just to express the fact that an increase in swimming speed during the nighttime could have significant effects on the migration of cells in the water column, in, for example, lakes or the ocean. So it might have that kind of effect and advantage. Another thing to notice is that the cells, so D measures the Chlamydomonas sinks at around 5 microns per second. So if you work out how long a cell would take to sink a meter, it would take about 50 hours. So individual cell sinking is not probably not a useful mechanism for going down the water column. And also I can show here the density of the cells that D measured, which was found to be higher during the night than during the day. But maybe not enough to explain the variation of sinking. And then another example where the speed might matter is Phototaxis. So here we have a similar model to before, but now we have a phototactic velocity, which depends on the intensity of the light, and also diffusivity, which depends on the intensity of the night. And in models that have been proposed of Phototaxis, the magnitude of the phototactic velocity scales like the swimming speed. And so if you have a swimming speed which changes when you've been culturing day and night, then you would expect to have potentially different results. For example, these patterns that were measured by Williams and Bees, they could be different if you were using different culturing conditions. So this is again a caveat about what culturing conditions we might use. And this is just for fun. So at time 0 here, you can see a light is switched on, and you have a suspension of Chlamydomonas reinhardtii, which had been settled to the bottom. And as the light is switched on, the Phototaxis causes the cells to swim upwards and animate in these bioconvection patterns. And then the cells have accumulated to the surface, and they form these plume instabilities. And then at some point, you're going to see the light switching off and everything just kind of sinking down. So it would be nice to actually grow some cells and image them at the same time to measure the motility parameters as the day-night cycles happened. which is not what we did. We sampled from cultures that were growing in day-night cycles. So it'd be nice to do a sort of live motility measurements. And again, for this situation, the culture conditions might give you a variation in the observed phenomenon. So this is a preprint that we just submitted with Praneet Prakash, which also deals with using light to concentrate cells above a porous medium, if you're interested. Okay, so I'll just conclude. So we found that day-night cycles strongly affect microscopic motility. So it'd be good to consider culturing conditions. And also this has consequences for observed macroscopic transport and flow. So in terms of the future, it'd be nice to repeat these measurements with differential holographic microscopy and differential dynamic microscopy to measure, for example, the flagella beat directly. It'd be good to test the hypothesis that I've been talking about regarding ATP availability. It's possible to measure ATP context using an expression using fluorescent dyes. It'd be nice to look at the effect of day-night cycles on chemotaxis and phototaxis. It'd be, as I just said, it'd be nice to look at the motility of a growing suspension, so directly, and also explore more about ecological consequences, such as Diel migrations in lakes and the behavior of Chlamydomonas in its native habitat of soil. I should also say it'd be nice to see how, so we had cells that were strongly synchronized, but it'd be nice to see how that synchronization is broken and how strongly the results depend on the synchronization. You know, so for example, does the swimming speed just go flat when you add tap to your media? Okay, so with that, I'll acknowledge my collaborators again, and I'll be happy to take any questions.